All right, today we're going to unpack the essential databases that power our understanding of proteins, the building blocks of life. Let's get right into it.
A protein's function is all about its shape. Its complex, three-dimensional structure dictates literally everything it does. So how do we study these incredibly intricate little machines on a computer? Well, that's where specialized databases come in. We need a way to not just store all this information, but to actually make sense of it all.
And that journey begins with the foundational library for all protein structures.
Okay, so first up is the Protein Databank, or PDB. This is basically the one global archive for every single experimentally determined 3D structure we have. Think of it like the world's public library, but for molecular shapes. Every time a scientist solves a new structure, they deposit it here, it gets a unique 4-character ID, and boom, it's freely available to everybody.
So how in the world do we get these detailed 3D pictures of something so tiny? Well, they aren't just guesses, that's for sure. They come from some seriously powerful experimental techniques. The big three are X-ray diffraction, nuclear magnetic resonance, or NMR, and the increasingly popular cryo-electron microscopy. Each method gives us a super precise snapshot of a molecule's atomic coordinates.
Okay, so you go to the PDB website and look up a structure. What are you actually looking at? Let's break down a typical entry.
Each entry is just packed with crucial info. You'll find the basics, like the molecule's name and what organism it came from. You'll also see which experimental method was used. But a really key piece of information is its resolution, measured in angstroms. Now, for methods like X-ray crystallography, here's what you need to remember. A lower number is better. It means a sharper, more accurate picture of the structure.
The PDB is absolutely incredible, but it is also massive, and it's growing exponentially. We're talking hundreds of thousands of structures. Just having them all stored in one place isn't enough. How can we possibly find patterns, compare them, and understand how they're all related?
And that is where classification databases come in. Two of the most influential are CATH and SCOP. Their job is to act like expert librarians, organizing that vast PDB archive into a meaningful system that we can actually navigate.
Let's start with CATH. It organizes proteins by looking at their core structural units, called domains. CATH uses a 4-level hierarchy to sort them out. It starts broad with class, which basically asks, is this domain made of mostly alpha helices, beta sheets, or a mix? Then it gets more specific with architecture, which is the overall 3D shape. Then there's topology, which describes how all those helices and sheets are actually wired together. And finally, it groups them into a homologous superfamily, which suggests they all share a common evolutionary ancestor.
Now, SCOP offers a very similar approach, grouping proteins into class, fold, superfamily, and family. What historically made SCOP so highly respected is that it relied heavily on manual curation. That means human experts would carefully review and classify the structures, creating a real gold standard resource for understanding protein relationships.
And this brings us to the most important distinction to understand. What's the fundamental difference between the PDB on one hand and databases like CATH and SCOP on the other?
It's actually pretty simple. The PDB is the archive. Think of it as the library that just holds all the books, the raw structural files. CATH and SCOP are the catalogs. They don't store the original books. Instead, they analyze them and tell you how they're all related. They help you find which ones belong to the same genre.
This table really highlights how they work together. PDB's type is an archive, and its primary use is to give you access to that raw experimental structure. CATH and SCOP are classification databases. They take that raw data and, using a mix of automation and expert input, allow you to do higher-level analysis, like studying protein domains or tracing evolutionary history. You really need both to get the full picture.
So ultimately, it's this powerful workflow. We take the raw data that we store in PDB, we use the systems in CATH and SCOP to classify it, and from that classification, we can achieve a much deeper understanding of biology. This workflow is absolutely essential for everything from designing new drugs to understanding the molecular basis of disease.